Welcome to the Rich Report, a podcast with news and information on high-performance computing. Today my guest is from Optalysys. We have Dr. Nick New. He is the CEO and founder of the company. Nick, welcome to the show today.
Hi, thank you very much.
Well, thanks for joining me all the way from the UK. You guys had some exciting news today, didn't you?
Yes, indeed. So we're just announcing the closure of our $4 million seed fund. And it's an exciting time for our company at the stage of technology development that we're at.
Yeah, and you guys are doing some pretty novel stuff with optical computing. So, Nick, I thought the best way to get this going is just go through your slide deck so we can get a better understanding, and then we'll do a Q&A at the end. That sound good?
Cool. Sounds good. Okay, so we are Optalysys. This company is the culmination of coming up to 20 years worth of work personally, starting from university days back at Cambridge University in the UK, doing a PhD on this technology, and we are close to bringing this technology to market now. So this is quite a late stage of development for us now. We are applying a well-established technology dating back to the mid-1960s using the powers of optical diffraction, similar to holography, to answer some of the big challenges that is facing computing today. And our vision is to revolutionize big data processing and model generation through our optical processing technology. And this comes at a time in computing where Moore's law is beginning to break down, which is well known. Computer chips are no longer getting any faster, and the power consumption of these very large computer systems is a real problem for people who have to run such systems. And our technology is very different. We are offering a very alternative approach to performing large-scale calculations of a certain type. And a key feature of our technology is that it's such low power. I'll talk a little bit about how our technology works. So our branch of optical processing is known as Fourier optics, the basis of which uses a diffractive optical process to produce two-dimensional Fourier transform calculations using parallel optical processing, low-power laser beams in conjunction with big advances from the displays industry in recent times, which has given us the interface into the optics. That's the electronic to optical interface that take the form of liquid crystal microdisplays, or to give them a technical name, spatial light modulators, which are basically postage stamp sized devices containing many millions of pixels. Now, the timing for this technology is an interesting point to discuss, in that the advances from the displays industry have led to very high resolution microdisplays being created for the purposes of projector technology. So this has led on from advances in the TV industry, leading to 4K TV displays, that has given us microdisplays that contain vast numbers of pixels. Now, those pixels we don't use as the basis of a display. We use them as a grid of numbers. So it is converting numerical information from electronics into this optical system via the modulation of a low-power laser beam. Big advantage of this process is the scalability. Now, the more pixels we have in our microdisplays, the more data points we have in our grids, does not reflect any increase in process time of that base Fourier transform calculation. And this same calculation, this same Fourier transform process, is used as the basis of very large scientific models, and can also be used in data processing for pattern recognition purposes in terms of optical correlation. So we compare these stages of Fourier transform processes together, but still keep that order one operation. And that's quite an exciting point to expand upon, in that we can have multiple stages of Fourier processing going on within one optical system without increasing that processing time again. So this offers a big scale up in terms of what we can do in terms of the size of the processing we can accomplish with one optical system. Just to expand on that, our sort of classic system architecture in this field is an optical correlator that does pattern recognition, and this is two stages of Fourier transform with a matrix multiply in the middle. So with this process we can compare images or patterns by displaying them on these liquid crystal microdisplays in an optical system. And then we use a CMOS camera sensor on the back end of this optical process to take the information out of the optical domain and back into the electrical domain. Now, this technology has existed predominantly in areas of the military in the past, but also on big optical tables within university labs predominantly. So we are taking a very different approach to this. We are forming this technology through our own designs into an optical co-processor technology that can then be inserted and integrated into standard computers or into a HPC rack, for example. And we're developing this with a PCI interface to allow users to actually plug this into their own computer and receive a boost in processing power for these certain types of calculations. So what we are developing is an optical system that is mounted upon a set of drive electronics, and that drive electronics can then be inserted into a standard computer to give that big turbo boost for certain types of processing. So the way this works is that we use multiple liquid crystal microdisplay devices. Low power laser light is projected through the system, and that converts the data represented on these liquid crystal pixels into an optical form that then goes through the optical system and produces this final calculation that's collected by a camera sensor that then feeds back into the computer. One interesting point to note is that we are developing this with certain applications in mind. So we are producing software wrappers and interfaces that allow this technology to be adapted to certain applications, but we're also going to develop this with a real open source feel to it to allow users to adapt and use their own data processing requirements to this technology. So the application areas that we are focusing on as a company, the potential application areas of this technology are widespread. So any areas which use very large amounts of data, areas which involve equation solving processes are applicable to this technology, and then this technology can offer advances in all these different fields within the scientific and industrial community. So we have chosen some key areas to work on to begin with, and I'll tell you about those in a bit. And we are focusing towards one sort of real commercial application to begin with. So this is the first focus of many different application areas that we shall be working on. As a company, we have been working with some key partners under some government and US government funded projects. Our main project has been funded under a UK government project with the Eartham Institute, which is a genomics centre in Norfolk, England. And I'll tell you about that, more about that in a second. We've also working with a consortium of 13 partners across Europe under a European Horizon 2020 project called ESCAPE, which is looking at the basis of the next generation weather forecasting models. And we've also recently completed a DARPA seedling project, so that's US government funded, looking at the basis of equation solving fundamentals under the Access program. So just on the DARPA project we recently completed, we've been looking at, in particular, how to extract the complex data from these Fourier transform operations as a real direct fit into the larger models using that single Fourier transform process that the sort of base optical function provides. And this includes taking what is usually an intensity output from the camera and using a part of our own IP of how to take the phase and amplitude from the signal at the end to give a complex readout. In terms of our ESCAPE project with the European Centre for Mid-Range Forecasting based in Reading, this is looking at breaking down the current weather forecasting models, which is using legacy code from around 35 years ago, and adapting it to the new types of hardware, new computer processors that exist in today's modern computing climate. And we are looking at what functions we can take from that model and apply with our optical processor. So our main commercial focus has been under a project codenamed GENESYS, and that's been in partnership with the Eartham Institute. As I mentioned, it's a genomics institute based in Norfolk, England. And this is relating to the alignment process of genetic data. And this is a process that is typically conducted on a HPC, and we've been looking at taking that and implementing it with this optical method. This has been a 2-year project funded by the UK government, which is close to being completed at the moment. And the main target of this project is to take a process that ran at 28 hours on their HPC down to 1.6 hours with our first optical processor system. And that's the other thing to mention in that is that the power consumption of that process, we believe, can be reduced by about 95% by switching to this optical process. And the other thing to mention with this is that the ability of this technology to be inserted into a standard desktop computer then takes away the need to have that HPC system there to be able to run this kind of alignment process as well. So it also changes the way that the scientists, the bioinformaticians, can approach these problems that they face. Just to describe the type of process this is. So looking at this genomics pipeline, the first stage being entering a DNA sample into a sequencer machine, such as an Illumina sequencer machine, results in many fragments of DNA code being produced. And those fragments need then aligning to a known sequence to basically piece together the new sequence of the material that you've put through the sequencer machine like a big jigsaw puzzle. This is very similar to a kind of needle in a haystack problem that optical correlation is extremely well suited to and has its roots in. So picking out small pieces of information from a very cluttered scene is what optical correlation is very good at. And this is what maps very well to this alignment process. And this is the first of several application areas within the genomics field that our technology can be put to. The way this works is that we take the base nucleotide information that comes out of the sequencer machine, and we convert that into a symbolic format that maps very well onto our spatial light modulators. We produce a binary format. This can run extremely quickly on a liquid crystal microdisplay, which typically has a frame rate of about 2.4 kilohertz. And then we map that into a correlation process. What that does is that every partial match between that part of code fragment against a frame of sequence data produces a spot of light, and that spot of light denotes the exact location of where that fragment has been located within that frame of sequence, and also, crucially, also gives a percentage figure of how well matched that fragment is. And that's of particular interest to the bioinformaticians, the geneticists, who run such a process called BLAST. And that's exactly what we're aiming our technology towards. What we've done in this project is we've proven that this technology can produce results that are as accurate as the very best software processors that the bioinformaticians use. And this is a very exciting development. We've been recently demonstrating this at an international conference down in Norwich in England. In terms of our system development, our technology development, for the first time recently, we've demonstrated this system in a coprocessor form that's mounted and integrated within a standard desktop format. So this was recently demonstrated at the Genome 10K conference in Norwich, showing for the first time how this technology can be formed in the future. So our next stage on from this is a commercialization stage. And to show you the system in action, this is an optical output you can see on the right-hand side, where matches, partial matches of a defined threshold are being located within a sequence, and then the exact location is passed on to a software process that produces the data in exactly the same format as bioinformaticians expect from an alignment process. And this is a crucial point to what we are developing. We are developing a very complementary technology in that the bioinformatician can send up the text files, the data files that he would normally use, and he would receive back a text file that contains exactly the same data in exactly the same format that he would expect from a normal HPC process. So he would not see this optical output, he would just receive his data back as you would expect, only in a faster time and taking much less power by doing so. So what this seed funding round allows us to do is move from the end of this project task and this stage of development that we're at and form a commercially viable system in the coming months. And it also allows us to focus on other areas as well, such as machine learning, where we are developing methods that are quite exciting in terms of convolutional neural networks. And we shall also be starting a Series A funding round in the next few weeks, as well as we look to scale up the commercialization and the activities of the company, both in the UK and predominantly in the US, where we also have an office in Reno, Nevada. Okay, that's the end of the slides. Thank you very much. If any questions?
Yes, absolutely, Nick. Thank you for that. It's fascinating technology. I wondered if you could go back to slide 18, just about your workflow as data moves through this from left to right. Are you starting with digital, converting it to analog, and then putting it back out to digital? Is that the flow?
That's exactly right, yes.
Yeah.
So this could be thought of as an analog processor with a digital wrapper.
Very slick. Okay. Yeah. Well, then I guess my next question is, like, you're taking a picture of a picture. How do you avoid losing resolution in that process?
Oh, well, in terms of avoiding resolution, well, the resolution of the system is actually down to the sort of pixel level of where the data falls on the camera. So it's we lose zero resolution through the system with this system we have here. The resolution is a key point to this. So a lot of work has gone into representing each base with as small a symbol size as possible, and that gives us a large degree of flexibility in terms of how much insertions and deletions and mutations we can fit into that data as well. So this gives us a very powerful and flexible approach to how that data is actually represented as it goes through the system. So in some processes you could be wanting to pull out exact matches. In others you could be looking at say very degraded DNA where the matches that you are looking for actually may only be 70% or less, for instance. And so we're developing methods that can pick out things at a very low level of match that normal software processors wouldn't be picking out.
Right, right. Well, FFTs are very widely used, as you mentioned, in HPC. And I'm wondering, as you've got this funding now, do you envision a way that people could test this over the cloud or something to see if it could benefit them?
Absolutely. So
Yeah.
we are just setting up a cloud offering at the moment, which will allow people to send up their data sets and for us to process them here and send them back in a report showing what the data is and how it's different to the normal alignment process.
Mm-hmm.
But in the coming months as well, we'll be announcing a full cloud offering where people can actually get their hands on this technology and use a cloud-based optical processor, as well as offering this as a units that can be purchased to go with their own computers and to bring that local level processing offering out into the field.
Yeah, yeah. Well, Nick, I guess kind of a wrap-up question. Could you envision a time when you shrink this down to some kind of chip package or something that we could just see as part of our laptops maybe someday?
Yes, absolutely. So
Yeah.
The size of the optical system, which is not a big footprint at the moment, so it already has the footprint of the standard graphics card.
Yeah.
But that can be reduced much further, actually. I mean, the liquid crystal microdisplay technology is shrinking as the months go by in terms of the pixel size of this thing. And the pixel size actually is one of the key factors in denoting how large these optical processors are. We can also shrink that by changing the wavelength of the light that is being used, the laser light being used through the system. So miniaturizing this further is definitely on our roadmap. And also integrating this system into other technologies as well. So not just computer systems as the kind of host.
Mm-hmm.
There's no reason why this couldn't go in the back of one of those sequencer machines I talked about before, but also in everyday life as well. So you could picture this as part of a machine learning or deep learning process through an AI in an autonomous vehicle, for instance. There's a wide range of application areas this technology could go into.
Well, Nick, this is fascinating, and congratulations on your funding this week. And
Thank you.
I want to thank you once again for coming on the show today.
Thank you very much. It's been my pleasure. Thank you.
You bet. Okay, folks, that's it for the Rich Report. Stay tuned for more news and information on high-performance computing.